but we see
Hi,
your screen. Yeah,
everybody. Yeah. Hi,
perfect.
everybody.
Yeah.
I'm Scott Chrisley. I'm co-chair with Jason Vanderheiden of the Data Representation Working Group, and I'm going to walk through the our report today. So data representation as a group is responsible for um, creating interoperability standards for AirSeq data, for sharing and analyzing that data. Much of the work that we do is very technical in nature. We write specifications for data formats, and we also write software to read, write, and validate those data formats. There's a GitHub repository where all of those specifications and software is available. And so our goals for the last year and a half are pretty straightforward. We previously had published a rearrangement TSV file format standard and schema. So much of our work has been doing community outreach and adoption and getting tools, analysis tools to adopt that standard. There's new standards that we've been working on, as well as refining those, those existing standards, the tools and the documentations. And as you've heard from Christian, and I'll repeat again in this talk, is that we do work with a lot of the other working groups to help them decide about their data models, how to represent that data, how to link it in relationally with other types of air data. So we do a lot of work with the other working groups, and as well as developing new standards. And I'll talk about that in more detail. So the community outreach, as I said, the main standard that we had was the rearrangement standard, which is sequence annotations and a file format for it. We released a paper in 2018, and on the left you can see that's the table of tools that were supported at the time of publication. We've been working to get new tools supporting it, and you can see that on the right. It's really great. We've added a number of new tools. I wanted to point out a few in this list. Cell Ranger. This is a tool that's provided by 10x Genomics. I think maybe you're familiar with them. They're a single cell technology company. So it's really great that we have a commercial provider who their own tool produces the air format. What you'll all see as well here are the IMGT tools. So it's been great. We've had a representative from IMGT join our meetings and provide input. And in this process of them adding support for the Air TSV format, they've also provided some improvements and enhancements to that. So that's really great. And the third thing I wanted to mention was that a number of these tools are part of the Imcantation tool suite. So what's really nice is now that whole tool suite throughout has support for the Air TSV. So that's great seeing that's a very popular tool suite. The other big effort that we did is to update the documentation website. There's a link here. I encourage you to go there. Note that this documentation website is not purely for computer programmers like myself. It's also for users. We've added a getting started section that has information about doing data submission for your AirSeq studies in relation to the MyAir standard. There's information about the Air Data Commons where you can query and download public AirSeq studies and the data associated with it, as well as many other resources, software tools, tutorials, examples, and some workflows. So, as well as on the documentation, we have details about all of the standards that have been defined. So, a lot of the work was to sort of encompass documentation from multiple working groups and include them together. And you can see a list, here. So, I encourage you to check that out and browse, and feel free to give us any feedback on that. Also included in that was refining the existing standards. So there was a new release of the Air standards version 1.3. This was primarily to include the official release of the Air Data Commons and also the associated repertoire metadata schema. So that repertoire metadata schema takes the MyAir standard, which says, here's what information you need to provide about your AirSeq study. And in this case, we make it, we formalize it into an actual file format that can be, that's interoperable and exchangeable. So, and there's many other sort of changes. I won't go through them in detail. Updates to the software packages as well that read and write these formats. So I encourage you to take a look at those if you're interested. As I say, we work with a lot of other working groups. And here's some of the things that we've done. One of the main things was the Data Commons API, which was in combination with the Common Repository Working Group. There's also the software certification, which was with the software working group, and then OgreDB. We've integrated to a certain degree in the documentation, and that came out of the Germline database working group. Okay, and then some new standards. So there's actually been work on this already for quite a while. So there are draft schemas that are available. So the rearrangement TSV was primarily annotations of sequence data. Now we're interested in further what I call analysis objects. So clones and lineage trees for B cells, the cell and receptor objects, which are going to be very important for single cell. So those are new standards that we're working on. So you've heard this from Christian. This is the exact same slide. I won't go through it in detail, but we're proposing to the community to merge with minimal standards and partially with Common Repository to have a new standards working group. We've actually been doing this for the last 6 months or so, and it's been working well. So I think a lot of us would enjoy that. Now, it does mean that our calls have been a little bit longer because we have more topics to cover, but everybody seems to feel that's an appropriate trade-off instead of having more meetings. And so our goals for the coming year is pretty much the same as what we've already been doing. So we're going to continue with our community outreach and adoption of the schema and the standards. Now we have more standards to sort of get tools and people to adopt. So we're going to continue that. There's continual refinement of those standards, the tools and documentation. I mentioned the clone and lineage type schemas, as well as single cell stuff. we continue to sort of take care of our role of working with other working groups to help them with their data models. And lastly, we have some sort of new kind of topic areas that we want to work with as we're increasing sort of the number of data types that we support. There's this idea of creating some sort of manifest so that you can package a series of files together and consider them a data set, something to that effect. So we're talking through ideas about that so that those things can be packaged together, as well as thinking a little bit of redesign of some parts of our schemas, in particularly the data and sample processing schemas, to make them a little bit more flexible. So that's some of the work that we have proposed. We encourage you to join us. Here's some links. You can email us. There's a documentation website and our GitHub where the specifications, the documentation, and the software resides. So thank you very much.
That's great. Thank you so much for that really amazing presentation and all the incredibly hard work that your group and the other groups have done to make all of this possible. It's really impressive to see the list of tools that are error compliant, and all of the upgrades in the standards and the documentation, especially that help people with their AIR data submissions. So really outstanding work. Do we have any comments or questions from the audience, from any of the attendees or other panelists? I'll give it a moment to look here. And panelists, if you see anything, or if people can also raise their hand, I think maybe. I'm not sure that you can do that as an attendee though. No, it looks like we're like everyone is okay. So again, we can, if you do have questions in the hot topics section, please feel free to speak up or write things into the Q&A.